Bismillahirrahmanirrahim. My name is Muhammad Hassan Islam, and today's my presentation topic is collagen biosynthesis.
Introduction. First of all, we need to know what is collagen. Collagen is a fibrous and insoluble protein that serves as the fundamental building block in the connective tissue of animals, including humans. Collagen is a structural protein found in connective tissues providing strength and support to skin, bones, tendons, ligaments, and cartilage.
The name collagen comes from the Greek word "kolla" meaning glue and suffix "gen" means producing. Collagen is the most abundant protein found in the human body. It makes up almost 25 to 35% of the protein content of whole body. Main fibrous components of skin are bones, tendons, ligaments, teeth, etc. Also influences cell shape, differentiations, and other cellular activities. And the collagen is also the main source of keratin. Basically, collagen is a structural protein found in connective tissues, providing strength and support to skin, bones, tendon, ligament, and cartilage. It is composed of amino acid forming a triple helix structure and is the most abundant protein in mammals. Collagen plays a crucial role in tissues integrity and health, with various types existing in different tissues. Like it is used in medical and cosmetics products. Now let's talk about the general structure of the collagen. Collagen molecule is rigid rod-like structure that stretching like stiff rod that stretches and has a complex structure known as quadrant structure. And collagen molecule is composed of three polypeptide chains twisted around each other, forming a triple helix similar to braided rope. And the polypeptides wrap around each other with the help of hydrogen bonding, which act like tiny glue dots holding the strands in place. Collagen is rich in glycine, proline, and hydroxyproline. Glycine occupies the third position in the amino acid sequence. And glycine is essential because any other large amino acid cannot fit in the center of triple helix structure. Here the sequence of amino acid is glycine XY, as you can see in the diagram. Now, so let's talk about collagen synthesis. It occurs inside as well as outside the cell. Now, which cell? Mainly fibroblast, that is the main connective tissue in the body. So, collagen is one of the proteins that function outside the cell, and polypeptide precursors of the collagen molecule are formed in fibroblasts, osteoblasts, and chondroblasts. These are secreted into the extracellular matrix. And fibroblasts are the primary cell responsible for collagen production in connective tissue. They produce secretion collagen fibers which provide structure and support to tissues such as skin, bone, and cartilage. And chondrocytes are cells found in cartilage tissues. They produce collagen fibers that provide structure and support to cartilage, connective tissue that cushions and support joints. Osteoblasts responsible for producing collagen in bone tissues and glycine, proline, and hydroxyproline to synthesize collagen fibers, which provide structures and support to bones. Now I will explain collagen synthesis by a diagram. For collagen synthesis, cells require nucleus, rough endoplasmic reticulum, and Golgi apparatus, and extracellular enzymes. It all starts from here. Nucleus have DNA and DNA have thousands of genes. In these thousand genes, 22 genes are for collagen production, and these 22 genes make 10 different collagen precursor proteins. So collagen gene is in the nucleus undergoes transcription and it produces messenger RNA. This baby RNA comes out of nucleus and here it is taken up by its partner ribosome. These ribosomes are attached on rough endoplasmic reticulum and here it undergoes translation and it produces a polypeptide which is known as pre-pro-alpha chain and it undergoes different modification while it is still being produced. like post-translation modification. I will tell about post-translation modification later, and these modifications are necessary while uh, when this pre-pro alpha chain enters into the lumen of a endoplasmic reticulum, and here is the N-terminal accessory. Amino acids code remove these accessory amino acids as a single molecule, and it helps in entry of this pre-pro alpha chain into the lumen of a endoplasmic reticulum. And that is why it is also known as single sequence peptide. After its removal, the remaining molecule is known as pro alpha chain, and it undergoes some modification. Number one, hydroxylation collagen has specific sequence of amino acids as glycine X and Y, as you can see in this diagram. Here X may be proline or lysine, and Y also with the initially proline and lysine that undergoes hydroxylation. Now it became hydroxyproline and hydroxylysine, as you can see in this diagram. The function of this enzyme is evident from this. Its name is not, and this hydroxylation helps in combining the fibrils of collagen, and how it works. Just wait a little bit. I will explain that. And most important, this reaction occurs in the presence of iron and vitamin C. Modification number 2, glycosylation cell have glucose and galactose. These glucose and sometimes galactose come together and get attached to hydroxylysine. Not all, just some of these. Now, what's the importance of this? So, such as assembly of collagen, fiber collagen, cross linkage, and collagen, and interaction with different other biomolecules. But all of these are just proposed theories. Now let's move to the next step, assembly and secretion after hydroxylation and glycosylation 3, which is triple helix formation of this. These pro-alpha chains come close and make interaction with the same chain and with neighboring chain. Now this we can call this interaction disulfide bridges. Now a molecule is formed and which we can call pro-collagen, and it have peptide on and C terminal, on N and C terminal. Now what is the importance of this? So it prevents premature degradation of pro-collagen. This pro-collagen now go to Golgi pattern and is ready for leaving the cell. So here is the end of intracellular collagen synthesis. Now let's start the extracellular collagen synthesis. After secretion, pro-peptides are removed, and it is done by an pro-collagen peptide and C-pro-collagen peptidase. Now the pro-collagen is called tropical collagen. Here collagen faces two fates. Either it can become long fibril, for example collagen type 1, 2 and 3, or it can make mesh network just like collagen 9 and 11. So we concern about fibrous not mesh network. Tropical collagen spontaneously combines and make an order overlapping parallel array with staggered pattern adjusted to neighbor by 3-4 molecule length, and now it's called collagen fibril. Did you understand? Now let me explain this. As you can see in this picture, it is the one tropical collagen, and this is another one. They combine in such a way that the leaves a gap of 3-4 of a molecule of collagen itself from its neighboring molecule, and it happens to all of molecule. This is happening in the 7 of the removal and C terminal domains. Uh, which is known as staggered pattern under electron microscope. A part of it seems black and the other seems white. Did you get that? Now the final step. Collagen fabric requires some power, and this is done by cross-linking formation. And as you can see in this diagram, number 8, cross-link formation. So here is the story. A minor group on lysine and hydroxylysine is got removed in the presence of lysine hydroxylase, and the remaining group, which is aldehyde group, come closer and combine with the minor group of another chain, and this result in condensation reaction, and a cross linkage is formed. Remember, this lysol oxidase is a copper containing enzyme. So this is end of the collagen synthesis, and in this way the collagen synthesis is performed. Now we can talk about post-translation modification of collagen. Post-translation modification involves the covalent and enzymatic modification of proteins. These modifications can occur at the C or N terminus of the protein and or the side chain of the amino acid present. Collagen undergoes post-translation modification after it is synthesized by fibroblasts in connective tissue. Post-translation modification of collagen are essential for its proper function. These modifications include hydroxylation, glycosylation, and cross-linking, which enhance collagen stability and functionality. Now we will talk about sources of collagen, which are animal products and plant-based food. Description: bones, broth, chicken, fish, soybeans are sources of collagen. Let's talk about some benefits of collagen. Basically, collagen improves the skin elasticity. How? It helps maintain skin firmness and elasticity, reducing the appearance of wrinkles and fine lines. It also helps in joint health. Collagen supports joint health by maintaining the integrity of cartilage and reducing joint pain and inflammation. It also helps in growth of strong and healthy hair and nails. It also helps to repair and strengthen the gut lining, improving digestion and nutrient absorption. Reduced wrinkles. Collagen supplementations can help to reduce the appearance of wrinkles and improve overall skin texture and tone. So this is all from my side. Thank you.